Understanding the physical basis of protein self-assembly in cells is limited by a lack of appropriate tools. Current methods suffer from low sensitivity, indirect readouts, limited throughput, and/or population-level resolution.
In contrast, our assay detects and quantifies the propensity of a protein for self-assembly in vivo with a sensitive, single-cell, high-throughput readout irrespective of protein localization or solubility.
When first trying the assay, getting the photoconversion right may require some empirical testing to obtain optimal conditions for achieving a good FRIT readout across an entire plate.
The sensitivity of the assay could also be compromised if the cytometer does not have separate detection filters for the FRET and acceptor signals.
Demonstrating the procedure with me will be Andrew Box, the lab manager of the Cytometry core.
To prepare a yeast culture for DAM-FRET, for every query protein, inoculate the transformed yeast colonies in triplicate. In 200 microliters of an appropriate non-inducing growth medium, in individual wells of a 96-well round bottom culture plate. Incubate the yeast cells with shaking with a 1.5 mm orbit at 1200 rotations per minute at 30 degrees Celsius for 16 hours. At the end of the incubation, sediment the yeast cells by centrifugation and remove the supernatants by forceful inversion. Add 200 microliters of an appropriate induction medium and return the cells to the shaking incubator for another 12 hours. At the end of the incubation, sediment the yeast cells by centrifugation and forceful inversion, then add 200 microliters of fresh induction medium. Then incubate the cells with shaking for